Hello, I'm Elizabeth Meinert-Spyker, and today I will be presenting my abstract titled Sickle Cell Disease Subtype and Sex Analysis of Diffusion Metrics on 7T MRI. This work was conducted in the Wood Neuro Research Group at CMU in collaboration with the University of Pittsburgh. The abstract focuses on understanding the effects of sickle cell disease, which predominantly affects people of African descent, often leads to neurological complications, and has been historically underfunded. Sickle cell disease is a genetic condition that results in the formation of an abnormal hemoglobin, HbS, leading to chronic hemolysis, anemia, abnormal perfusion, and decreased oxygen delivery to various tissues. Here you can see a schematic of how sickle cell disease affects red blood cells. The left image shows red blood cells with normal hemoglobin, or HbA, and the right image shows the sickled red blood cells that are produced by HbS. This video shows how sickled red blood cells can block blood vessels. There are multiple forms of sickle cell disease, with HBSS, HBSC, and HBS beta-positive thalassemia being included in this study. As neurological function is correlated and reliant on adequate blood and oxygen delivery, it is common for neurological complications to arise in patients with sickle cell disease. To analyze how sickle cell disease affects the brain, our collaborators at the University of Pittsburgh began collecting 7T MRI data in 2017 on patients with sickle cell disease and age and race matched controls. In this abstract, I analyzed the diffusion tensor imaging data that our collaborators collected. DTI is a method for characterizing microstructural changes in the brain and can be used to evaluate disease progression and treatment effectiveness. Several metrics can be obtained from these images, with fractional anisotropy being the most common and mean axial and radial diffusivity being other DTI metrics analyzed in this study. These DTI metrics are based on eigenvalues or tensors, which can be visualized in the image on the right. To the best of our knowledge, this is the first study to use 7T MRI data to compare diffusion metrics between various subtypes of sickle cell disease. We hypothesized that fractional anisotropy and axial diffusivity would be significantly decreased in patients with HBSS compared to mild sickle cell disease, while radial diffusivity and mean diffusivity would be increased. We expected to see these same trends when comparing severe and mild sickle cell disease to healthy controls, with differences being more pronounced in patients with severe sickle cell disease. For the comparison of sexes, we hypothesized that males would have significantly increased FA and significantly decreased RD in both controls and patients with sickle cell disease. In my analysis, I included 24 healthy controls and 24 patients with sickle cell disease. Of those with sickle cell disease, 11 had HBSS, 9 HBSC, and 4 HBS beta-positive thalassemia. The average age of subjects was 36, and there were no significant differences between the age and sex of the controls in patients with sickle cell disease. DTI data and T1-weighted MP-RAGE scans were acquired with a Siemens Magnetome 7T MRI scanner and a customized 16 or 32 channel head coil. I performed pre-processing of the data with the softwares MRTrix and FSL. Tract-based spatial statistics was used to compare diffusion metrics between groups. Voxel-wise statistics was then conducted by creating design files which corrected for sex and age and analyzed the differences between controls and patients with mild and severe sickle cell disease. There were no significant differences between DTI metrics of patients with severe sickle cell disease and patients with mild sickle cell disease. Various brain regions of patients with severe and mild sickle cell disease were significantly different from healthy controls. This can be visualized in the image on the left, where the top two panels illustrate the comparison of controls versus severe sickle cell disease, and the bottom two panels indicate controls versus mild sickle cell disease. Blue to light blue values indicate that individuals with sickle cell disease have significantly lower values than controls, while red to yellow pixels indicate that individuals with sickle cell disease have higher values than controls. Results indicate decreased FA and AD values in patients with severe sickle cell disease compared to healthy controls, and decreased FA and increased RD in patients with mild sickle cell disease compared to controls. Comparison of sexes found that AD was lower in healthy males compared to healthy females, but found no significant differences in patients with sickle cell disease. This may indicate that sickle cell disease affects the sexes differently. In conclusion, our results are indicative of white matter tissue damage in patients with sickle cell disease. To the best of our knowledge, this is the first study to use 7T DTI in adult patients with sickle cell disease. Analyzing the effects of sickle cell disease of the aging brain is essential to develop and analyze treatment methods, and we hope that DTI can be used as a method to characterize treatment effectiveness in the future. Future studies will classify patients as severe or mild based on phenotype expressions instead of genotype. Here are my references. Thank you very much for listening, and if you have any questions, please feel free to contact me.